Okay. If you have any questions, feel free to interrupt and just say something, because I'm not sure I can see your hand signals. Okay. All right, so let's start with this. In biology, we often are attracted to biology because of the great biodiversity on Earth. And so, the classic case is Darwin's finches. So sometime, many thousands, well, much more, many years ago, a finch made it to the Galapagos Islands, and then the finches moved to different islands, and even on the same island, they developed different beak shapes. And the idea of these beak shapes is they can eat different things. So, for example, this one is kind of thick and it's good for eating fruit. And this one is kind of pointy and it's good for eating insects. All right. And so I think a lot of us are drawn to biology because of this biodiversity. And related to biodiversity is genetic diversity. So often we think of genetic diversity within one species, but you have this diversity that eventually gives you biodiversities in different species. Okay. And this is related to natural selection in that we select for the best genes in some ways. And this relies on the fact that we have genetic diversity so that we can select for things. And so here's another example that we all probably know. It's a textbook example of the moth, right? So this is a dark moth, and this is the original spotted moth. It's called the pepper moth. And before humans, before Industrial Revolution, the peppered moth was good for the tree trunks because the tree trunks were usually more or less white. But then humans started burning coal, and the result is that the trees became darker. And so then the original peppered moth has a disadvantage on this tree because the birds can eat it. And so when a mutation occurred that gave us a dark-colored moth, they are selectively favored in this case. And so this is a type of natural selection under action. And, so this is a textbook example, but it happens to every organism all around us, all the time. Okay, so here's a slightly better picture. I don't know how well you can see it on the screen, but this is the peppered version, and this is the dark version. All right. This is just to show natural selection in action. The y-axis is the dark form, the melanic form. All right. And so since about 1959, there have been some scientists looking at the frequency of the dark form, and it's dropped over time. And this is because somewhere around here, in the US and elsewhere, we have laws that reduce the amount of pollution that comes out of our factories. So with less and less pollution, the trees are less and less dark. And so it would slowly favor the peppered form over the dark form. Okay? And so this is a positive human action. We've reduced our pollution. It's good for our own health, but it's also good for the peppered version of the moth. Okay? And so this is selection. You select for the pepper, the lighter form over time. Okay. So for people like me, we're interested in the genetic basis for how this happens. What gene, if it's simple, one gene maybe, or many genes underlie this color difference? All right. So a common approach is to do something called genetic mapping. You try to map this phenotype to a position on the chromosome. And because you know the position on the chromosome, you can find the gene that controls it. And then once you know the gene, maybe you can understand the molecular biology behind the phenotype. So for example, in this case, being dark. All right. To do this, you need many markers. And there have been many types of genetic markers, but nowadays, the focus is on single nucleotide polymorphisms. Okay, so that's short, SNPs. is the abbreviation for single nucleotide polymorphism. And this is the dominant type of marker that we're using. Okay, so how do we map? Here's a cartoon. So here you have a heterozygous fly, okay? So the top is the chromosome from the father, for example, and all the alleles are orange. So each spot is a genetic marker along this chromosome. And then it's heterozygous, so the opposite chromosome is from the mother. In this case, we draw it all as white, okay? All right, and so this means there's a difference between the father's copy and the mother's copy, okay? All right, so in the heterozygous individual, it's completely heterozygous, but in the next generation, there will be recombination, meiotic recombination. And so, recombination happens more or less randomly at different parts of the chromosome. So maybe you get a recombinant chromosome that recombine here. And so, this is the father's allele, this is the mother's allele. And then some other one has a different one, where the recombination is here, and here, and here, and here, and so on, okay? All right, so then we have some measure, some way of determining the genotype at all these locations. And I'll tell you about rad sequencing later, but you can imagine any other method to know the genotype at this position, at this position, at this position, and so on. We also have the phenotype. So we can look at the phenotype. This is by the asterisk. So this individual has the phenotype. And so in this case, maybe it's the eye is a little bit orange. Drosophila eyes are usually red. All right? And so this other one is also orange, but these are plus. Plus means it's normal. So they're probably normally red, okay? So you're looking for orange eyes versus red eyes. And these three all have red eyes. These two have orange eyes. So this means, and we know the orange eyes come from the father. And so if we have the father's allele here, then we know it must be from here to the right? Because here we have the father's allele, but it's not orange. So we know that these are not correct. None of these are possible for where the location of the gene is. We can also compare here. Both of these are red, but, orange, the phenotype orange, and only these two are the important ones that are still associated with the phenotype. So we can exclude these two. So genetic mapping basically means we look at many individuals, we genotype across the chromosomes, and then we look for regions that make sense. So in this case, this region makes sense. This means the mutation is somewhere here. Okay, and so that's kind of driven by this picture here. All right, so that's the general concept of genetic mapping. I'll show you a second example. This one is, again, you have two different strains. So this is not so important. These are nematodes, but you have strain 1 and strain 2, right? So it's, strain 1 is homozygous for this. Strain 2 is homozygous for the orange. And here we have a mutation. Okay, and so the mutation can be anything that we care about. So the worm, the nematode, maybe moves funny, okay? All right, so you cross them, and you get the F1. Now, here, the right-hand side, this is where the mutation is. We don't know it right now, but this is where the real mutation is, on this chromosome. The left-hand side is another chromosome. So imagine this is chromosome 1 and this is chromosome 2. Chromosome 1 does not have a mutation. We don't know it today, but that's the answer. Chromosome 2 does have a mutation. Okay, again, we don't know it, but through mapping, we will find out it's on chromosome 2. All right, so you have your F1. Again, it's completely heterozygous for all the chromosomes. And then you let recombination happen. So they have children, or they cross and have children. And you have many different recombinant chromosomes. So, for example, this individual got blue on the left and orange on the right. This one actually didn't have any recombination, and this one had orange and then blue, and so on. Okay? All right. So when you pick, here they're picking mutant animals. Okay? So here's the mutation. We don't know what's here, but we do know what it looks like. It looks like a funny worm. Okay? So we pick the funny worms. Because we picked the funny worm, it must be homozygous where the mutation is. And so, therefore, on the mutation chromosome, it must be this way. All right. So we need to genotype these individuals. So individual A, individual B, and C, and D, and E, and so on, okay? All right. And so imagine we have many of these, and so that's kind of like what this is. So each line is an individual. And we pick them as mutant, so they must have a mutation on chromosome 2. On chromosome 1, we don't know it, but the mutation is not here. And so how do we tell the difference? We look at the genotype. All right, let me pause here for a second. So here, conceptually, let's say at this location, it's either A or G. All right, so you see the orange, that means it's A at this location, and then it's G at this location. Okay? All right. So again, because we pick mutants, it's 100% has that allele, has that mutation here on chromosome 2. On chromosome 1, it has nothing to do with the mutation, so it's random. So in general, you should have a half chance of getting either side. So then it's 50%. All right? So anywhere on chromosome 1, on average, you have 50% of the blue allele and 50% of the orange allele. Whereas on chromosome 2, where the mutation is 100% the blue allele. And then farther to the left and farther to the right, it slowly goes back to 50-50. All right? And so this is the general idea of genetic mapping. Is everyone okay so far?
I have a question, Prof.
Yes. Go ahead.
The blue represents the mutation?
No, technically blue represents the first strain.
Okay.
The red, it's a diamond. Go ahead.
In unlike chromosome, there will be no mutation. So what is 50%?
50% of either the orange allele or 50% of the blue allele.
Okay.
But they can be different. Yeah. Because here it's A's and G's, but maybe here it's C's and T's, maybe. Is that okay?
Yeah, okay, thank you so much.
Okay. All right. All right. So if we go back to this moth case, they did something like this, and they mapped it to this black region. So on the moth chromosome 17, they did that analysis, and they know it's here. And they call it, the melanic, they call it carbonary. All right. And then this was an early study, and then later they were able to find out. So this black area is here, okay? So you see the B and you see the D. That corresponds to here and to here. All right. But if they sequence more carefully, they find out that the pepper moth is missing a segment, whereas the dark form has an extra piece of DNA. And they found out that this is a transposon. So I don't know if you know what transposons are, but they're so-called jumping genes. Sometimes they're called parasites in the genome. These are pieces of DNA that can move and copy themselves in the genome. So the dark form had a transposon that jumped into this gene, and this change causes it to be a dark, okay? And so the key is they use genetic mapping to map to this location, and then they use more advanced genetic mapping to find out the exact mutation. So now we know why we have the peppered form and the dark form. All right. So this is just long introduction into why we care about RAD sequencing. All right, so the notes are here. That's an organism that you read in a textbook, or maybe you care about, but it's unlikely that you're actually studying that organism. You might be studying your favorite insect or plant or microorganism, mammal, whatever, okay? And but if we want to do some type of analysis comparing interesting phenotypes, we have to do genetic mapping. And this means we need many markers. And again, most of the time, these are SNPs. So the question is, how do we get them? And so that would be the RAD sequence that I'll tell you about soon. And then the second point I want to make is the pepper moth color is a so-called simple phenotype. So it's a color change, and it's only caused by one gene, or the difference in one gene creates the darkness versus the normal pepper phenotype, right? And this will be different from when I talk about a supergene. Okay. So now it's the genomic era. So it's really easy to sequence all or most of a genome. And the price is not free, but it's getting cheaper and cheaper. So there are many ways to get markers. And the three dominant ones, they fall into these three classes. So the first one is called reduced representation sequencing. This includes RAD sequencing, and I'll tell you about that in a minute. A second approach is so-called low coverage sequencing. This means we try to sequence the organism, but we don't care too much. We try to get some data, but not all data. And the third way is we do care exactly how the sequence is. So we sequence whole genome resequencing. So we sequence a lot of that individual, and we make sure we think we know exactly what the DNA sequence is. All right. So today we're not going to talk about these two, partly because they cost more money, and partly because this way is quite powerful for many biological questions. Okay. So reduced representation sequencing actually is a group of multiple ways of looking at things. And these are four. There are more, but these are the four dominant ones. The first one is RAD sequencing, which I'll tell you about. The second one is exon capture. So, you know, in eukaryotes, the genes expressed from an mRNA, and the mRNA is often spliced because there's the exons that are important for protein coding, but there's also the introns, which aren't really interesting. We don't have to sequence the whole genome, we just sequence the exons. And there's techniques to capture the exons. A third way that's been used quite a bit in biodiversity research is ultra-conserved elements. And so these are pieces of DNA in the genome that stay the same in many, many organisms, sometimes for unknown reasons. So we can design ways to capture this. And so this is another way of sequencing important information without having to sequence the whole genome. Another way is transcriptome analysis. This is straight RNA sequencing. So you want to look at how the RNA is expressed. At the same time, you get the sequence. So then you can see if the transcriptome is different between two individuals. All right. And so we're not talking about the bottom three, but we'll focus on RAD-seq today. Okay. RAD-seq is a type of genotype being by sequencing, and this will become clear in the next slides. RAD-seq stands for this, restriction site associated DNA marker sequencing. Okay, so the RAD is the restriction site A associated DNA, and then RAD-seq is this sequencing. All right, and so as you can maybe guess from the name, restriction site is where the restriction enzymes cut. So here is a cartoon of a chromosome, and you cut with a restriction enzyme. And there's a couple of choices, but let's say you choose a common one, EcoR1. So it'll cut every time there's an EcoR1 site. And then the next step is you add this thing called an adapter. And this adapter allows you to sequence this part right here. All right? So the whole point of this is you cut and you sequence this side and this side. And you'll notice that you only sequence this part, so which means you're not sequencing this part, and you're not sequencing the middle part. And maybe you sequence here, but you're not sequencing this. And this saves you a lot of money. All right. So this is the very basic way. And a way to make it even better is to add something called a barcode. And so that's what this little green thing is. So again, we cut the DNA, but this time, instead of attaching only the adapter, we attach the adapter plus the barcode. And so barcode looks like this. So you have individual 1, you purify the DNA, you add the adapter and barcode. You have individual 2, you add an adapter and a different barcode. So the individual A barcode in this case is GCCGT. Okay, and then the barcode for number 2, individual B, is AATTG. All right, so Illumina, we have the primer on the blue part, and you sequence the green plus the orange. The green is the barcode, so if you see this, then you know that comes from individual A. You see this, it comes from B, and something else will come from individual C, and so on. And then you sequence the orange part. So I don't know if you can see it, but it's the GAT, CGA, TCCGA. And then at this point is the SNP, is the variation. Okay, so it's A in this individual, and it's T in this individual. So now we know the genotype of this person, this one, and this one, right? So again, this is genotyping by synthesis. So you synthesize, genotyping by sequencing. You sequence this, and you simultaneously get whether it's an A or a T at the SNP that we care about. So that's the whole premise. Now I'll go through some more details. So this is a cartoon of the pipeline. So as mentioned on the previous slide, we cut with an adapter. So it cuts at various places, okay? And then we ligate the adapters. That's what these orange things are. So it should be on both sides. All right, and then there's a couple of steps that are for molecular biology. And so if you ever do it, we can talk about it. But you have to, you can mix samples because we have barcodes. We break up the DNA a little bit, and we choose good sizes to sequence. And these are all molecular biology reasons. And then 6, 7, and 8 are similar. We do some repairing, we add A tails, and then we ligate the Y adapter. So these are just things that make sequencing easy and efficient. And that the result is you get this thing. And then Illumina, we run it through the machine and we sequence it. So we'll sequence on this side and this side of each read. So the left side and the right side. And again, this is where the restriction enzyme cut, and this is where the restriction enzyme cut. Okay, so here's a zoom in of that. Again, the gray is the DNA that you cut. So it's cut here, and it was cut here. And we ligate the primer plus the barcode. All right, and then we pool the samples together. So the different colors are different barcodes. So here, you might have 10 different individuals. One of them's purple, green, blue, and yellow, and so on. And so this part saves money because you can do everything at once. All right, and then we have to add this so-called Y adapter, which is this white thing. It's designed so you don't get PCR from this guy, but you only get PCR from ones that have a true adapter. Okay. All right, and then this is the result that you get, and this is what will be sequenced. And so this is the Illumina barcode, primer sequence and barcode, and then you sequence this part. So again, you only sequence part of it. And then this part is unknown. And you can also sequence this part here, 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 and here. Okay? All right. And so this is what it really looks like after you compare it to the genome. So on top is your genome. These are restriction sites. And you get these sequences right next to the restriction site. And so here, you have 10 or 15 reads, 10 or 15 reads. Each line is a read. And this is different from trying to sequence the whole genome. when you sequence the whole genome, it's supposed to be random, but sometimes you miss some spots, and sometimes you get some spots with a little bit more. But you spend the same amount of money, and here you're very confident of whatever the genotype is here and here. But here you're not so confident because you only have 1, 2, or 3 reads. And so you're always worried that there's a mistake somewhere. And this makes you worried about your analysis. So RADseq lets you be very confident about this and this. The trade-off is that you don't know any information here. But often information here and here across the whole genome, because the restriction site is found in many places, you get thousands of pieces of information on the genome. And that's often enough to do a lot of biodiversity or ecology or evolution questions. Okay, so that was a walkthrough of the protocol. All right. So there are several varieties of RADseq, and they come by many names. So one of them is EasyRad, 2BRad, blah, blah, blah. And right now, this thing called DDRad is probably the most popular. DDRad stands for Double Digest Rad. Okay, so here on top is what I just described to you. Again, we cut here and we sequence to the left and to the right. We cut here, sequence to the left and right, and so on. Double digest rad, you actually cut twice. So you cut with a restriction enzyme that cuts on the black marks and a restriction enzyme that cuts on the gray marks. And you attempt to get the fragments that are a mix of black and gray. Right? But there's a size element to it. So if the black and gray are too close, this is thrown away. So you never see data from here. Here, on this side, it's too long, so you don't see data. But on this side, you can. All right? The big advantage of double digest rad is you cut it with the second enzyme, and that gives you usually more reliable fragments and a little bit less number to look at. So it's easier in many things than regular rad sequencing. Okay. So in that paper I passed around, it talks about several variants, and so here are some of them. And so there's this rad sequencing is called GBS, but it's still the same idea. EasyRad or DDRad, and you do various different things. And if you need to do it, you can look at it and try to understand what's going on. They just differ in slightly different ways in hopes of getting slightly different. It's easier to get data or faster to get data, or people want slightly different data. Okay. So every technique works on paper, but it may not always work in your lab. And so there's some important errors and biases to RADseq data that we should think about. The first one is the so-called dropout allele. And so that's what the top part is trying to say. When you cut, so this enzyme can cut on the left, and it can cut here. So on individual 1 or haplotype 1, this DNA fragment only cuts here and then way far over here. And then, but on haplotype 2, you have a cut here, cut in the middle, and a cut here. All right. Remember, we put some adapters on, and somehow we get a certain size. And this size might be too big. Whereas this size is okay. You get this size and this size. So you have sequence on this side, here, here, and here. Whereas here, this fragment is just too long. It never gets into the machine. So here, you'll get information, but here you'll get no information. So for individual 1 or haplotype 1, you've lost information. And depending on how you're doing your analysis, you may not know that. So this is something you have to think about when you're analyzing the data. A second issue is this thing called PCR duplicates. And so the authors divided into original RAD. That's the one I walked you through, all of the details. And then some of the newer ones. And when you do original red, you cut, that's where the red mark is, but this side is random. And it's really hard to get the same random length. So when you do PCR, you might have more than one copy of this one, this one, this one, and this one. And so in this cartoon, you have 3 copies of the circle, 6 copies of the star, 1 copy of the triangle, and no copies of the square, okay? But you can tell you have 3 copies because they have exactly the same length. And this is really rare when you do original red, which you break the fragments randomly. So the net result is you have 3 independent reads. And the 4th one you lost because you never knew it. But you do not count this 3 times, and you do not count this 6 times because you know these are artifacts of duplication. Now, in the other techniques, basically you're always looking between 2 restriction enzymes. So if you get a read on this side and you get a read on this side, even though they're coming from different individuals, they have the same length. So you have no idea they're coming from different individuals. And so here again, you have 3, 6, and 1, but you don't know these 3 are duplicates, and you don't know these 6 are duplicates. So you end up saying you have 3, 6, and 1. And sometimes that's important, but sometimes it's very, sometimes it's not important, which is okay, but sometimes it is very important. And this has similar problems, right? So my point here is that RAD-seq is pretty powerful, but you have to be aware of certain problems. Okay, so I said DD RAD-seq is probably the most popular right now. I told you this part, PCR duplicates are unknowable, so that's a weakness. But the benefit is it's easier to tune and has better reproducibility. Okay, so tuning, what does this mean? This means how many markers per genome. And these are some analyses you can do with SNP data. When you have lots of sequence, you can do these things. So the easiest one for us might be phylogeny. You have sequence from humans and chimpanzees, and they should be more similar to each other than compared to birds, for example. All right, so how much sequence do you need? You don't need that much. And so you can design DD-RAD to produce some data. So maybe 100 sequences per animal, per individual. Or you can get more and more and more and more. So a study called population genomic scans, which I won't talk about, but it needs lots and lots of markers. And so you can tune DD-RAD to have a lot more. And so this is this thing called tunability. You can use restriction enzyme 1 and 2 that gives you a few markers, but maybe use restriction enzyme 3 and 4 that allows you to have much more results. Okay, and then better reproducibility is this idea that because you're cutting twice, you always know the size, and so it's easier to get the same data for all your individuals. Okay, how are we doing so far? Any questions? Okay. So I'll move on to how my lab uses RAD sequencing to solve a problem. Part of my lab works on fire ants. And so this is a picture of the queen in the middle. And these are workers. And this is a monogyne colony. It has 1 queen, mono for 1. Okay, so there's 1 queen in this entire nest. In this colony, it's polygyne, so it has many queens. So each one that has a paint mark is a queen for this colony. And so you see workers, and if your eyes are really good, you can see the brood, the larvae of the colony here. All right, so this is a fundamental difference. It's the same exact species, but it has a single queen form and a multiple queen form. And we had 2 goals. We wanted to build a genetic map, which I'll tell you in a little bit. And by building this, we wanted to test for this thing called a supergene, which I'll also explain in a little bit. All right. So again, this is our fire ants. We grow them in the lab like this. So these are big plastic containers. And then this piece is a Petri plate. The Petri plate has plaster. And so when you open the Petri plate, it looks like this. And so this is the queen in the middle. And this is one of a monogyne colony with lots and lots of workers. Fire ants, we care about it for several reasons, but one of them is because it's an invasive species. It was native to South America, and it invaded into the US on the eastern, southeastern side, and then later from there, it has moved to many countries, including my country, Taiwan. Okay. A lot of times we actually don't care about insects too much, except when they cause a problem, and fire ants are a problem. So this is the fire ant abdomen, the gaster, and this is its stinger. And it will sting you when it attacks, and it'll give you poison. And this poison is injected into an unlucky person. So this person, every time you see a white pustule is where this person was stung by a fire ant and got injected by poison. And typically after 2 or 3 days, you develop these big white itchy pustules, and that's very uncomfortable. And so because of this, we actually care about it because humans like to be happy. All right. And so fire ants can kill. So some people have allergic reactions to it. And so every year, a few individuals are unfortunate. All right. So they impact human health. They make our lawns and our yards they don't look good. This is a colony. This is a small colony, but it makes your green grass not so green. It affects agriculture, so it stings us, but it'll sting other farm animals. And all of this creates a lot of problems. So you spend lots of money trying to fix this. Okay. So again, this is our system. The question is single queen versus multiple queen. And this is a cartoon. So this is a single queen colony with the big one is the queen, and these are the workers. And this is a polygyne colony. So several queens and then workers. And what I want to emphasize with this slide is that there's many differences beyond just queen number. Okay, so one of them is this. So if you look in the field, if all the colonies are monogyne, you'll find that the colonies are far apart. They're far apart because the workers from colony 1 and colony 2 will fight. So each one is a little country. They fight and they protect the area, so then they are far apart. Whereas in the polygyne case, if you have a field and you see lots of colonies that are very close, that's because the workers don't fight. So actually, colony to colony, workers can move back and forth. All right. And so how does this form? In part because in a monogyne colony, each colony is founded by a single queen. She flies and lands and digs a hole and makes a colony. In polygyne, it's a little bit different. She flies, she mates in the air. So on fire ants, the males and the virgin queens have wings. They fly, they mate while flying. But then the polygyne queens make a decision and they join back to the nest. It can be the same nest or a different nest. All right, so then this nest gets more and more queens. And at some point, the nest gets too big, and they decide to split off. So some queens and workers decide to move neighboring area to make a new colony. All right. So the queen sizes are a little bit different. What I mean by that is if you look at this picture, the head and the thorax are the same size, but this is the abdomen, something called the gaster also. The monogyne queen can lay many, many eggs. So this is full of ovarials and eggs that can be laid. So she can lay hundreds to thousands of eggs per hour. This is the polygyne queen, and her gaster is much smaller. And so she's less fecund, she has less ovarials, less ovaries, and so the result is she cannot lay as many eggs. So if you look at a monogyne, the real queen has a big abdomen, whereas the polygyne ones have small eggs. And another characteristic is, on average, the workers in a monogyne colony are a little bit bigger than the polygynes. All right? So, take-home message is, polygyne, in addition to have several queens, have different phenotypes. Some of it's behavior, some of it is the product of behavior. They do different types of making new colonies. Their worker size and queen sizes are different, right? And the monogyne has kind of the opposite, all right? So there are many, many different things that are different between a monogyne and a polygyne, even though it's the same species. Okay, so almost 30 years ago, they found out that this was fully linked to a gene GP9. So this was from basic genetic mapping, and they thought at that time it's GP9, but the basic mapping was very basic, so it doesn't have enough information. All right. Another interesting aspect of fire ants is it has this thing called a selfish locus, or selfish gene, or selfish behavior. So in a monogyne colony, if you look at this gene GP9, it has two alleles, a big B and a small b. But in the monogyne, everyone is big B, big B. So I draw them all as black, okay? So the queen is big B at this gene GP9, and also all the workers. In the polygyne colony, if you look at it, the queens are all big B, little b. All the queens, every single one is big B, little b. The workers, there's two types, big B, little b by the blue ones, and the black ones are just like monogyne, big B, okay? So here, the difference in queen number is incredibly separated by the queens have very different genotypes, and this is important. All right, so a monogyne, if you do some behavior experiments, you find out that they stay single queen because they kill everyone. So if a big B, little b queen tries to come into this colony, she gets killed. A big B queen normally tries to make its own colony, but let's say it's lazy and it tries to come in, it also gets killed. Here, it's different. They will kill all the big B ones, but they will keep the big B, little b ones. And more importantly, it's this worker that seems to decide. The big B, little b worker apparently accepts this and does the execution. And this is different from the big B workers, which apparently don't do much. So that's why we call it a selfish gene. This worker is big B, little b. It sees this as little b. It selfishly accepts this queen. This worker looks at this one and notices that there's no little b and kills it. All right, so at the gene level, little b recognizes little b and helps little b. This is something called a green beard. Green beard is this toy example that this author, scientist Richard Dawkins, wrote. And the idea is, let's say there is a gene and it gives you a green beard. But this green beard is a magic green beard. You only see it if you have a green beard. So you have a different colored beard, you don't know they have green beards. All right, and then next to the green beard is this behavior locus where you help other individuals with a green beard. And so now you have this thing where the phenotype, the beard, and the behavior are linked together. So for our fire ants, this would be somehow little b maybe has an odor, a smell, that they get recognized. But everyone else that does not have a green beard doesn't know this at all. Okay. So then this brings us to the fundamental question, or the big puzzle we had at the time. How can there be so many phenotypes, but only controlled by one gene, one locus? So again, remember, there's many differences from here compared to here. But it seems like only one gene controls it. Maybe we don't actually see it as one gene. Maybe it's something more complicated. And therefore, for this reason and others, we thought that GP9 is a marker on a supergene. A supergene is this thing. It's many loci, many gene locus. It acts as one piece of DNA, but it has many genes where they can co-evolve. And the idea is that there's no recombination or little recombination separating the genes. So in humans, we have an X and Y chromosome. The Y chromosome and X chromosome are kind of like two different alleles of supergenes because there's basically no recombination between the X and Y chromosome. So all the genes on the Y chromosome accumulate mutations together, and those typically help males. Whereas on the X, because it's found mostly in females, it might have genes important for females. All right, so this was our model at that time. So this is GP9. This is a chromosome and has GP9. And so let's say GP9 is here, and it's the big B. So we call the whole chromosome the big B allele or haplotype. And then this is the little b one. So on little b, it's the same gene, right? But it's little b. And let's say there's an inversion. An inversion actually reduces recombination. So remember, in meiosis, there's recombination between the chromosomes. If there's an inversion, it's actually hard to get recombination here. You can get recombination to the left and to the right, but here it's much less. And the result is this group of genes stay together. Okay, so here I'm drawing a cartoon of 4 genes, GP9 plus 3 other genes, and they always stay together. This red one never shuffles with this one. And so if this one gets a mutation, it's a big B mutation, and if this one gets a mutation, it becomes a little b mutation. So you get a bunch of little b mutations and a bunch of big B mutations. And you can imagine that all the big B ones are good for monogyne, and all the little b ones are good for polygyne. I also draw these things, the transposons, they're jumping genes, and for theoretical reasons, there should be more transposons on the little b allele. And so this is what a supergene is. You have this block of DNA. It has many genes. I draw 4, but in fire ants, there's like 500. And all of them, they have very little chance for recombination because there's a chromosome inversion. Okay, so back to our goal. That was our model. So if we build a genetic map, we can test for this inversion. All right, so what we did is we take a big B, little b queen, heterozygous queen, and then we get the males, we isolate DNA and do rad sequencing. Okay, so that's what I told you before. And then we do genetic analysis. And part of that's done on the computer because there's a lot of sequence data. Here's some not important things for today, but we had 2,000, almost 3,000 markers that we could use across the chromosomes to look for, to build a genetic map and see if there's something funny happening at GP9. And software gives us this 16 linkage groups. So there's 16 chromosomes in fire ants. So this is a very satisfying result. Okay. So let me walk you through a little bit of bioinformatics. This is a queen, queen's diploid. So, you know, one comes from the father, one comes from the mother, and there's recombination. So you can imagine this individual has blue on top and pink on bottom. This has a mix, and so on, okay? We take the DNA, and then we add the Illumina adapters, and so this one has barcode AAA, AGAGT, and so on. And then we sequence this on the Illumina platform, and then now we have data. Once we get the data, the first thing we have to do is split by barcode. So in this case, GGAGG is this individual. So we capture all the individuals that have GGAGG, and we know it comes from this one. And we do the same for this one, and this one, and so on, okay? So that means bioinformatically, we look at the sequence, we look at the first 5 nucleotides, and we assign it to the individual. All right, the next step is to get rid of the barcode, because this barcode is artificial. It's not part of the genome, so you cut it off. The next step is this thing called quality trimming. Quality trimming means how good the sequence is. You keep the good stuff and you remove the bad stuff. And so the Illumina machine will give you, an A here, and also give you a quality, like how good it thinks it is. And so these are all good, 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 good. And then at some point, we think it's bad. So we chop it off. So everything after 63, in this example, we don't want. But that's for this read. And every different read, each read might have different cuts. It might be cut at 63, it might be cut at 50, it might be cut at 75. All right. When I was doing the analysis, I got a little lazier, and I just said, well, it also has to be at least 50 base pairs. So some of the quality trimming might have cut it to short, and the short ones I didn't want. So I said, you have to have 50. And then I did this thing called merging identical. So if everything's the same, it has the same information. So I don't know how much bioinformatics you've done, but if you want to map this read to the genome and you have 10 copies, you have to map it 10 times. And that takes computation time. But it's actually easier if I make all 10 of them one, and I say there's 10 of these. And so that's what this was. And then I map it to the genome. That's what number 6 is. Right. And then after that, I did some post-processing. I made a database, and then I made the linkage map using this software, MST map. All right. So at the time I did this, I wrote all of this software because for many reasons. But I wrote it myself, and that's actually not the best thing to do. The better thing to do is to find software that can already do it. And so today, a lot of people use this software package called Stacks. And Stacks basically does what I did, plus a few other things. Stacks is not the only software. There's these other ones. So you have 6 or more choices to analyze RAD sequencing data now. Okay? All right. So when you analyze the data and use MSTP map, the software to make genetic maps, you get this type of drawing. And each little line is a locus or more than one locus. And so this is linkage group 1, and there's all of these markers across the chromosome. And we can get this genetic map. So this is chromosome 1, 2, and so on. And I told you that we know this gene GP9. And so GP9 is found right here. And what's special about it is this, even though it looks like one line, there's actually hundreds of markers here. Right? And so here's another way to look at it. This is the same chromosome 16. It has 265 markers that have not recombined in our family. And that is really rare. Everywhere else, we only have 1 or 2 markers that don't recombine. But here, this is 10-fold more. And so we know something special is happening. If we investigate more carefully, it looks like this. These markers corresponds to 12 megabases of sequence. Whereas all of these corresponds to 8 megabases. So there's a lot of recombination here, no recombination here, and then some recombination at the bottom. And so this is a hallmark of a supergene because there's a non-recombining region, meaning the big B genes and alleles can evolve together on big B, and the little b can evolve together on little b. All right, here's another way to look at it. So if we expand it all, this is the non-recombining region. And GP9 is found here, and so there's almost 500 more genes inside this red area. And conceptually, it looks like this on a chromosome. So A22 is here. This part is freely recombining, but the green and the blue are here and here. And so on the big B, it's this order, but on the little b, it's inverted. And so here is a fish picture. It's not super clear, but this is the chromosome 16. You see red, green, and then blue. In this chromosome, it's red, blue, and then green. So we have low recombining, and it's also because of a chromosome inversion. and that is this fire ant super gene. All right. How are we doing so far?
Prof, I have a question.
Yes.
So you said that the big B and small p gene is kind of selfish. That if a queen from big P will come in their colony, they will kill it. So why are big P and big P worker ants in that colony? I mean, why there are not only big P, small P ants? Yeah, here.
Yeah. They kill queens, but they don't kill workers. And we don't know why. I mean, we have a guess. Like this queen, we have some data that says it smells different than this queen. But there's no real data that says that this worker and this worker smells differently. So they think they're sisters. They think they're friendly, family members. Whereas here, they make a distinction. And they smell nice, and this smells bad, and they kill this one. Does that answer your question?
Why workers in big P, I mean monozyte colony kill their queens?
Oh, that's because a monozyte colony is what I call a standard colony. A standard colony is you don't want anyone else besides your own mother. So even though it's big B, the strategy is kill everyone that's not your mother. Because in an ant colony, most of the time you want to help your relatives. You don't want to help someone else's children because it doesn't help your genetic fitness in having future progeny. Yeah. Does that make sense?
Yeah, thank you.
Okay, thank you. So I will continue. Sorry. How long is this class supposed to be? I can't hear you.
I guess 2 hours.
Oh, 2 hours? Okay. All right. So I told you about a fire ant social supergene. Sometimes we call it a social chromosome because it's most of the chromosome. And at the beginning, people just kind of laughed at us. Not really, but kind of. Said, well, this is just a strange thing in fire ants. It has nothing to do with my organism. It has nothing to do with other organisms. So the question becomes, is it rare or common? Well, it turns out, I will show you a bunch of stuff, and it's not rare at all. And you might argue it's kind of common. All right, so there's this ant called Formica silesi. So this is it's a different ant. It's a type of wood ant. So you'll find it high up in the mountains or in the high latitudes, so northern parts of Europe and Asia. They used, fire ants, we use rat-seq using families. So that's exactly what I talked to you about. In Formica silesi, this other research group, this ant also has monogyne and polygyne forms. They used GBS. So GBS is just a type of rat sequencing. They also built a linkage map like we did, so this part's exactly the same. But then they did something called looking at LD, linkage disequilibrium, across the supergene. This is a fancy term for, they looked at many families, and they see if this supergene looks like a supergene still. Their key finding is that it does have a supergene, and it's on a different chromosome. So that's like if we find another mammal and they have XY sex determination, but it's actually a chromosome 1 instead of our human XY. So it looks XY. Here it looks like a supergene, big B, little b, but it's a totally different chromosome. Different chromosomes have different genes. And that's pretty cool because that means it's convergent at the supergene type, but the genes are different. Okay, and so this is what our paper that we'll talk about at the end, okay? So I won't talk more about this one right now. Now, this is Formica silesi. This research group, they looked at other Formica, and they found that many Formica species have this supergene. So this is where the supergene is. It's on their chromosome 3. And when you see a drawing like this where it's really high here, that's an indication that there's some type of supergene happening. Okay? So like here, you might see something high, but it's not very wide. So we would not call this a supergene. But here, you have this thing that's high and wide. We typically call that a supergene. All right. The point of this analysis is that there's this supergene has been found in its population 20 to 40 million years ago. So the original ancestral species divided and separated and evolved into these different species. And our best estimate is that this happened many, many millions of years ago, which means the supergene has been there for a long, long time. Okay, so that's, sorry. This says Formica silesi is a second independent case. In the genus, there's many, and it looks like the same one. So two different ones in fire ants and then in Formica. Now, there's even another one. Cataglyphis is a different type of ant. It also has monogyne and polygyne, and they also found a supergene. And so this is the chromosomes, and again, you're looking for high area that's wide. And so on their chromosome 2, you see this big peak. This is the zoom in of it, so this is the presumptive supergene on their chromosome 2. All right. But the unique thing, the interesting thing about this one is this is their chromosome. So this is their chromosome 2 compared to our chromosome 16. This part is the same. So this is the same chromosome. They call it 2, but it's really the same as our chromosome, my fire ant chromosome. So that means all of the genes in the green area, of course, corresponds to the blue area. These are the same genes, or nearly the same genes, that are involved in the supergene. This means this convergent evolution at the chromosomal level. This suggests that there's some genes in here that are often used to make you polygyne or make you monogyne. And that's kind of interesting too. That's like if in mammals we find another XY animal, but it became XY differently, but it's still the same chromosome. So it's like completely independent, but convergently you find the same chromosome used. All right. And so here is a simplified phylogeny of ants. The red one is the fire ant. And it belongs to this clade. And then this is, the green one is the current species, and the blue one is Formica, what I showed you earlier. They belong to a different clade. Okay? So this group, the top group and the bottom group are separated by a long, long time in evolution. And so this clearly is a case where Cataglyphis and Solenopsis are using the same chromosome to do single queen versus multiple queens. It's actually genetically closer to the Formica species, but they use different chromosomes and different genes for social organization. So that's pretty cool because you have independent and you have convergent. And that suggests the powerfulness of biology. And this might tell us something unique and special about this chromosome that might be repeatedly used for single queen versus multiple queen biology. Okay. There are many more social chromosomes in ants. You're getting more and more. Some of them are not published yet, but these are two examples that are published. So this is an ant called Pogonomyrmex californicus. It's this ant here. It's a so-called seed harvester ant. And they have a different version of monogyny and polygyny. When the queen makes a nest, it has two choices. it can make it independently, that's the classical monogyne, or it can group together and make a polygyne colony kind of like as a teamwork. And so this difference is associated with a supergene. This one is a different ant. It's called Oceroburoi. That's not so important, but they have this worker. They're mostly workers. It's a clonal ant. It's a weird ant. And it's lost its queens. But if there's a mutation in a part of it, you actually get the queen back. And every time it does this, it's a big piece of DNA. And so it's consistent with the supergene controlling queen versus worker. And so it's not monogyne polygyne, but it's queen versus worker. And it appears to be a big supergene also. And there's a couple others I haven't talked about. Okay, so those are ants. Bees also seem to have one. So these are bees in Africa, and there's the so-called Scutellata subspecies and the Monticola subspecies. So this one's found more in the mountains. This one's found in the lowlands. And if you try to understand what's different between them, they seem to have two possible supergenes. So here's a map again. These are the different chromosomes, and there's a high peak here on chromosome 7, and another high peak on on 9. It's not as wide, but it is not small. And so if we zoom in on 7, you see a bunch of stuff here, and you zoom in on 9, there's a bunch of stuff here. So again, there's tens to hundreds of genes that are linked together, presumably for the mountain phenotype or for the lowland phenotype. All right, so those are social insects. But you can clearly find them in other insects. One of them is in the butterfly. So Heliconus is another textbook example of Mullerian mimicry, where I always forget. Mullerian, I think, is one is poisonous and the rest are not poisonous, and they try to mimic the poison to get not eaten. And Pepio is a different butterfly, and they have Batesian, and this is where everyone is poisonous, and they try to mimic each other. All right, this is a so-called supergene of genes. So this one is regulated by many genes, kind of like in the fire ant case. This one is a supergene of regulatory elements. What this means is you have a gene that's expressed, and there's enhancers or promoters, and many of those enhancers or promoters are functioning together as a mini supergene. Okay, and so here's the case in butterflies. So here are lots of different morphs, and this is the model and this is the mimic. So this one mimics this one mimics this one, approximately, and so on. Right, and here's a different one. It's the same species, Heliconus, but genus, but it doesn't have as many differences. And it turns out that there is a supergene here that is important for these wing pattern differences. All right. So those were from almost 10 years ago. We might say, well, it's less and less fashionable. But just last week, there's a new paper talking about stick insects. And so Refugio is a location in California, but these stick insects have two types. The so-called green one, which is almost all pure green, and the striped one, which, if you look carefully, has a white stripe down the middle. All right. In this difference, so they can mate. It's the same species. It turns out, and there's two populations that they know about. The scientists are studying. There's probably many, many more. And so mountain one, the Refugio mountain, has this striped form and the green form. And the second mountain, about 30 kilometers away, also has the same, a striped form and the green form. When they do genetic analysis, hopefully you start to understand. Again, they look at it and you see this peak here, and then so on chromosome 8 is a supergene. And so this is a zoom of chromosome 8, and you see lots of points that are high up, indicative of difference between the two morphs, the green one and the striped one. So this is their supergene. It's a little more complicated, and I'm not sure this is the easiest way to see it, but they have So refugio, they have the stripe sequence. So they sequence this one, and this one. So they have 4 sequences. So the stripe one and the green one from the same mountain. Green one and the stripe one from the same mountain. Okay? And so the supergene between the stripe and the green is this area. So there's been a movement of DNA over here and a movement of DNA over here, and there's no recombination here. And then in the other mountain, there's also a recombination event, but it's not as big. So in this mountain, there's a green in the stripe, and the major difference is here. There's another one here that's not important for this model, but here it is important. All right. And so it's 2 independent movements of chromosomes, a piece moving and then a piece moving, and a piece moving and a piece moving. But it turns out there's a little bit of overlap. And so they think the important genes are focused around here. Okay? And so again, this is a case of independent evolution of the same thing through chromosome changes, possibly using the same genes. All right. So those are insects. And you might say, okay, well, those are crazy stuff for insects. Nothing would ever happen to vertebrates, but there are. And this is one of my favorite ones. So this is it's called a ruff, and it has 3 morphs. So you have this independent morph. So it's this guy or this guy. They're both independent. And then you have these satellite ones. So they have about the same size, but they're all white. Okay, so these 2 are almost the same size, but one has lots of color and one is all basically white. Then you have this 3rd morph called a fader, and this is a male, but it looks very much like a female. So it's small, it looks like a female, it's kind of cryptic. And how they mate is the independence. This one's independent, it's moving its wings, it's moving its body, so it's making itself look very beautiful and large and powerful. And then the females really like this. So the females end up mating with the independents. And the satellites, they're almost the same size and they hang out, and they kind of dance and move with them. And so even though the independent mates more, they sometimes get some matings because they're next to the powerful people. All right. The feeders, they're kind of sad. They cannot do this dance because they just don't look charismatic enough. What they do is they mix with the females and they sneak copulations. So they try to trick the females into mating with them. And then they look a little bit like females. So then these males don't attack the fader type. And so you have 3 different mating strategies. And so it's polymorphic. There's lots of different changes to the body size, the coloration. There's also physiology changes. And so they wondered if it was a supergene, and they found it. Okay, so this is a bunch of phenotype differences. We don't have time to talk about it, but on here the axis is going down, and then on this chromosome there's a huge region, and this part is where the supergene is. So the ruffs have it. This is another bird. It's a sparrow. This one has a crazy mating system, and so how does this work? So you have a white morph, so it's white on top, and then you have a tan morph, it's tan here. Oh, so it's white here and on top, and tan here and on bottom. And the tan males like to mate with the white females. The tan females like to mate with the white males, and vice versa. So the tan ones don't like to mate with tan. So there's rare female-male matings here, and there's rare female matings here. So it's kind of like 4 sexes. You have to have a white male mating with a tan female. But the white almost never happens. And then you have to have a tan male mating with a white female. But this one never happens, or rarely happens. So to ensure the whole population, you have to have all 4 morphs to make it. And then if you analyze it, it turns out there's a multiple inversion supergene. So like the black, blue, and green areas have been moved around, and this creates supergene linkage in this region for this mating. I'll skip this in the interest of time. And then just a couple years ago, there's this quail one. So the quail, they have a lighter coloration to darker coloration. Again, there's a super gene on chromosome 1. So super genes are everywhere. I talked about the birds. I talked about the butterflies and my ants. But I didn't talk to you about fish, but they have some. I didn't talk to you about mice. This is more about a selfish gene, like a true selfish gene, but they have super genes. And lots of plants, depending on some of their mating preferences and such, is also regulated by super genes. So super genes are everywhere. All right, so here's a summary of today's lecture. RAD sequencing. I introduced this technology, restriction site associated DNA sequencing. So you sequence next to the restriction sites. This is a way to identify many markers, SNPs. You actually don't need the genome to know this. You just do it and you get sequences. And then using that sequence, you can do lots of biological analyses. You only sequence part of the genome. You ignore the rest. This is usually enough information for a lot of analyses, and you don't have to spend your money sequencing everything. And so that's why it's relatively economical. Molecular biology is not hard, but you do need to be careful. So as long as you're careful and you learned a few steps and you get it working, you can apply RAD-seq to almost any animal or plant or microorganism you care about. Okay. It's a powerful tool. I talked to you about a lot of stuff, and it falls under the framework of population genomics, because you're looking at the genome, not the complete genome, because we're doing RAD sequencing, but we're looking at many individuals. Sometimes it's a family, sometimes it's many different individuals. And in the examples I gave you, I didn't tell you, but some of the cases are true populations. They just they take random samples, right? And through this, we can try to answer ecology, evolution, biodiversity, and other applied questions. And so I didn't talk about it, but here are some things. What genes are under selection? So these can be speciation genes or domestication genes. So this is like an applied aspect. This is basic biology. And so this is kind of what we did talk about with respect to the supergenes, social supergenes. We can ask, has selection been acting, or can drift be an explanation? This is kind of esoteric, so I'll skip it for today. What genes contribute to a trait? So at the beginning, we talked about the pepper moth, but in applied direction, we care about agriculturally important genes. So you can use RAD sequencing to help you map genes that make you have more rice or taller crops or whatever. You can also use the information to look at past demographic history. So, for us, we might be interested in human migration. Humans came out of Africa and then they moved around the world. And so when did this happen? What was the population sizes and so on? And we can infer a lot of this from sequence data. Often with conservation biology, we might be asking, is it one big population or many small ones? So climate change is probably going to make the Earth hotter in most places. So you can imagine a species moving from the lowlands to the mountains. So then each mountain becomes like a virtual island, right? So then you become one big population to a small one. And then if you do the analysis of data, you can ask, does this really happen at the genetic level? All right. If this is a complicated slide, the point is I'll send you some of the slides. And these slides, this is like what you can do with genetic data. So when you get RAD sequencing data or any other type of genetic data, you can do all sorts of things. So just to highlight one, we made the linkage map in fire ants. You can do other things like test for genotype association. So this is like the moth case, the pepper moth case, okay? Or demography, migration rates, population sizes. So like, where did humans come from? How big were the population sizes? Or, maybe you don't care about humans. We can talk about, an ant population or an endangered species that's found somewhere. All right. Then on the supergene side, I talked to you about the fire ant supergene. We call it a social supergene because it affects single queen versus multiple queen one. Simultaneously, it's a selfish supergene. We were the first ones to find a social supergene. But social supergenes have been found in many other ants. One example is the independent evolution of supergeneness. So this is the paper we'll talk about. It's different from the fire ant. Another case is convergent evolution on the same chromosome. So that's one of the examples I gave you with Cataglyphis. And then I listed some supergenes for other social ants. Okay. All right. Supergenes, which are often caused by inversions, are likely common in nature. I think we just have to look. And so I gave you examples from insects and birds, but in that summary slide, this one, you have it in many, many different types. And so they're likely everywhere. So probably the most important take-home messages are here. Supergenes are probably the basis for many complex phenotypes and polymorphisms. So if you have an organism that has two morphs, it's very complicated differences. I would say there's a good chance it's actually by a supergene. It probably contributes to both cryptic and obvious speciation and biodiversity. And so in Drosophila flies and in the mosquitoes, there's quite a few inversions of chromosomes. So basically they're supergenes, and they have slightly different characteristics. And so that will slowly lead these to different, the flies to different flies and mosquitoes to different mosquitoes. All right. So that's the lecture. Before we talk about the ant paper. Any questions or comments here?
Ada pertanyaan. Wah anjing, apa cuma? Nggak ngerti apa-apa.
Mia, are you speaking to me? If you are, I cannot hear you. If you're not speaking to me, then that's okay.
Yes, Prof. I have a question.
Yes?
As a student in agricultural science, we learn about ecology and behavior of agriculture insect. Could you please explain how supergene influence insect behavior or ecological adaptation?
Okay. Let's say you have an insect that can, well, like on the sticky insect example, the striped morph and the green morph, they prefer different host plants because they can survive on the host plants better. So let's say there is a pest species that attacks a certain crop better or worse, and then there's another morph that's in the forest. So like, because of this polymorphism in morph, one of them might stay in the forest, but the other one might be better on human crops. And so that might mean there's a refugia in the forest if the forest can maintain the polymorphism. So like maybe it's a heterozygote in the forest. Yeah, and so I don't fully know because I haven't really looked into the agriculture aspect, but if you see two different morphs, then that might be the case. Now, of course, humans are using lots of pesticides, and so you might have a pesticide-resistant one and a normal one. Those are probably caused by single genes. But if you see big differences, one possibility that it's a supergene. Now, whether you can control it or not, because you know it's a supergene, that's still a little unclear right now. Okay, any other questions?
Okay, thank you, Prof. But I have one question again.
Yes?
If a chemical or insecticide affect supergain, does the impact show up in the short term or over a longer period?
Say again the first part. Did you say insecticide?
Yeah. If a chemical or insecticide affect a supergain, does the impact show up in the short term or over a longer period?
Let me see if I understand your question. You're saying, like, let's say there's a chemical that affects the big bee and fire ants, but not the little bee. Is that your question?
Oh. Can the chemical or insecticide affect in a supergain, Prof?
But you mean like differently, like it affects one allele, like big B different from small b. Is that your question?
Yes.
I can only say maybe. What I mean is, so in the fire ant,
Yes, I mean, like chemical or insecticide can affect supergain, like can make a mutation or something.
Oh. The most likely answer is no, because DNA is just DNA. So it's not likely that you will affect the gene, the super gene. But in the super gene might be a receptor, and that receptor might be sensitive to the chemical. So let's say in fire ants, on the big B version, there's a receptor that's sensitive to a chemical. You can use it to kill the big B individuals, but the little b individuals might be resistant because they might not have the receptor. So if you're asking about mutations into the super gene, it's probably not possible because that's usually random. But if you're asking if a gene in the super gene makes a receptor or makes something that is sensitive to the chemical on one haplotype, but not on the other haplotype, then that's possible that you select for one version. Good?
Yes, thank you very much, Prof.
Okay. Any other questions or comments from the other people? Okay, then let's move on to the paper. And I'm hoping you can help talk a little bit. All right. So this is the paper that I asked you to read. Can someone tell me what is happening in figure 1A?
Wah, oh, nggak bisa Pak, sungguh-sungguh tapi nggak bisa tuh. Saya dulu, sungguh jauh banget jauh tuh.
Should I call on someone? How about Elkanah? I don't know if I say your name right. Are you here? Can you give me your best guess as to what's happening on figure 1A. I see people are escaping because you are afraid to answer. All right. Someone, a brave volunteer?
Wah, aku nggak isi, Cok. Sumpah.
Someone else? Oh, Marietta. Any thoughts? What does the blue and the green mean? What's the difference?
Wang lanjai.
How many blue ones are there?
27.
Marietta, I think you can answer that. Count it. How many blue boxes are there?
6.
Maybe 16.
Right, green boxes.
The green is 27.
Excellent. So what do you think they represent? If I tell you fire ants have 16 chromosomes, what do you think is happening?
Apa ya, Cok? Oh.
And the other species, Formica, has 27 chromosomes. What do you think each box represents?
Wah, aku nggak iso, Cok. Sorry, cannot. Cok. Sumpah susah ya, aku untuk
Which color do you think are the fire ants, and which color do you think are the other ant?
The fire ant is the blue one,
Good, excellent.
and the other ant is the green.
Nice. Okay. Can you guess what the we call them ribbons? The lines. Can you guess what these lines are?
These are linkage groups.
Explain more.
linkage up.
The box itself is a linkage group. So like the 1 and the 2 and the 3, that corresponds to the linkage group. So chromosome 1, chromosome 2, chromosome 3. But you see this. What do you think this means? Chromosome 3, and then you see a bunch of lines. Some of them go here, some of them go here, some of them go here. And this one, it goes to this one, it goes to this one, and it goes to this one.
These are markers
Yes? But why are there lines?
on where they can find differences.
Yes, good. But what does it mean? What are the differences meaning? Or the green 3 is darker. The box, right? It also has all those green lines coming. What do you think chromosome 3 is? on the other species? The fire ant has a super gene, and the other species has a super gene. Where do you think the super gene on the other ant is?
I think the super gene of the fire ant is the 16, and the other ant is number 3.
That's right. Good. Right? And so what do you think the lines mean? Or what is the author trying to tell us using the lines?
Tinang di Cok, hilang konto. I see.
Are they the same chromosome in the two species?
Baik kung ito ka pa naman.
The lines connect the same chromosomes. So is chromosome 3 in the other species and chromosome 16 in the fire ant the same chromosome?
Yeah, I mean, in fire ant, the chromosome 16 is linked with the other species chromosome 5.
Okay, good. So then, are the super genes on the same chromosome?
Sorry, I didn't understand.
Are the super genes on the same homologous chromosomes? So you said the fire ant is on chromosome 16. It's mostly chromosome 5 in the other ant. So is chromosome 5 the super gene on the other ant? Where's the super gene in the other ant again?
It's on 3.
Okay. So is the 3 the same as 16?
No.
Excellent. That's the key, right? The whole point of this figure is to say that chromosome 16 is the fire ant super gene. Chromosome 3 is the other ant, the alpine silver ant. That's their super gene. But if you ask where the homolog is, they don't match at all. And so this is this independent evolution. And for evolutionary biologists, it's a very cool result because different ways give you the same apparent phenotype. It's a little bit like mammals have X and Y sex determination. Drosophila flies, the fruit fly, also has XY sex determination, but it's clearly not the same chromosomes. So at the chromosome level, it looks the same. It looks the same because it looks like there's an X and Y in Drosophila and an X and Y in humans, but it's not the same set of stuff. It's not the same genes. And so here it's the same idea. Chromosome 16 for fire ants, there's the big B and the little b. That creates monogyne and polygyne, whereas it's chromosome 3 here. They don't call it, they call it M and P. And it's not the same chromosome. And that's powerful evolutionary biology in action. Okay, excellent. The rest of the stuff is more complicated. We don't have to go through it. But I'll take questions. If you have any questions about this paper or comments about this paper. Was the paper easy to read? Hard to read? No one's saying anything. I'm assuming it's easy to read?
No,
I
it
think
was
that's
very
right.
complex, but we
Okay.
somehow understand.
Okay. That's good. I mean, to read, you have to, it takes time. You have to push yourself a little bit. Okay. So let's see. Yes. Sorry. Rotel.
Yeah, sir,
Go
I
ahead.
have a question.
Yes.
Yeah, so I mean, what's the main difference between the social chromosome in Formica salici and the Solenopsis invicta?
They're two different chromosomes. So the chromosomes have different genes. Well, let me say it in a different way. You can imagine like an odor, a smell-producing gene for queens. And if you smell one way, it's monogyne, and you smell a different way, it's polygyne. And you can say, well, maybe every time there's a single queen and multiple queen colony, it's the same odor, the same smell gene. But they found a different chromosome. So that means it's not the same genes. And so it's a big mystery right now. What are the important genes in Solenopsis invicta? And what are the important genes in Formica salici? Our guesses are that there's some odor genes, some receptor genes, probably some neuron behavior genes that make them behave a little bit differently. There might be metabolism genes, and that's all we know today. All we can say is that it's not the same toolbox, if you want to call it that. So chromosome 16 in fire ants has 500 genes. That's like a tiny toolkit, like a toolbox of 500 genes. And Formica salici is using another set of 600 or so genes that are another toolbox. It's different metabolism genes, different receptors, different whatever. Does that make sense?
Yes, sir, thank you.
Thank you. Any other questions?
I have a question, Prof.
Yes, go ahead.
So the scent in workers and pheromone in workers and the queen is totally different, or they have same smell?
Oh. In general ant biology, the colony has a smell, but the queen and workers also smell differently. So like if you do chemical analysis of the queen and chemical analysis of the workers, you'll find that they have different cuticular hydrocarbons, and they often have different other chemicals. But cuticular hydrocarbons is what most people study. It doesn't mean that's the only thing that's different, because it's easy to study cuticular hydrocarbons. Some of the other chemicals are hard to study. But yeah, there's the queen just smells different from the worker, but somehow the queens and workers in one colony have a similar smell. So colony to colony, the two colonies have slightly different smells. So you can know it's a different colony.
Thank you, Prof.
Any other thoughts or questions? Yes? Go ahead.
I want to ask about, it was previously explained that in ants, some worker ants are killed because they are different. Does this also happen in the other social insects? Maybe like bees?
Oh. In almost all social insects, they will kill workers from a different colony. So bees will kill bees from other colonies. Wasps will do the same. Termites will do the same. Yeah, so in general, they protect their colony. I mean, there's always a few exceptions, and there's some bees that can sneak into another colony, so they somehow make it, and they somehow cheat and make it in and like steal food or whatever. But that's like an evolutionary arms race. But yeah, in general, they try to keep everyone out. It's kind of like passport control.
So I have a question related to that. Like, so if in case of fire ants, so work from big P colony, if worker ants enter in big P small P colony, so the workers will keep them or kill them.
Kill. Yeah. Because that one's based on colony odor. The polygyne will accept other polygyne workers. Sometimes we call that a super colony. But yeah, monogyne into polygyne will get killed. Okay. I have a question for you. How many are master students and how many are undergraduates? Are there any undergraduates? All masters?
There is one student in undergraduate.
I see. Okay. And you all have a master's project that you have to do?
Not yet, sir.
But you have one for a requirement to graduate? Is it a requirement to have a project to graduate, or you can just take classes?
Yes.
Okay.
We need to do that.
Okay. Okay, I think that's about it. Mia, do I need to do anything else? Otherwise, you see me next week and the week after. And next week I will give you another paper, 1 or 2 papers to read, okay? All right. I have another question. What were the topics in your previous lectures?
Previous lectures in bachelor's?
In this class, in insect molecular biology.
In seven weeks, we more learning about basic biomolecular. So,
So did you learn about mutagenesis? No?
No, not that.
Okay. What about transgenics?
Transgenic. Belum juga sih.
Okay, so next week then I will talk about mutagenesis and transgenics, okay?
Mitogenesis dan transgenik.
Okay. And then I will think of something for the 3rd week, but I will ask you next week, because if I teach you something that's already been taught, maybe it's not so useful. Okay, thank you very much. Have a good day.
Okay, thank you so much, Prof.
Thank you.
Thank you so
Bye-bye.
much, Prof.
Wah, sumpah, Cok. Real penipuan kok. Cok.